Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And good evening, ladies and gentlemen. Welcome back. It is a great pleasure to have you return to this conference. Has been quite a day so far. Now, it is a time for the long-awaited moment. We are pleased to invite our second keynote speaker, Professor Dr. Tunku Kamarul Zaman Tunku Zainol Abidin, the Professor of Orthopaedic, University Malaya, to present to us an interesting topic titled Orthopedic Implant Designs, Current Trends and Future Development. Before that, allow me to introduce briefly about him. Our speaker is by profession an orthopedic surgeon with special interest in upper limb and microsurgery. He graduated with a medical doctorate from the National University of Malaysia in 1998. After serving the Ministry of Health as a medical officer for several years, he entered the Master of Orthopedic Specialty Training Program from University of Malaya and graduated in 2004. He also received a diploma in tissue banking from the National University of Singapore in 2003 during his specialist training. Professor Dr. Tunku Kamarul Zaman, Tunku Zainol Abidin, then pursued his sub-specialty training in University of Liverpool, UK from 2005 until 2009 and was also awarded a PhD degree from this university. Professor Dr. Tunku Kamarul Zaman, Tunku Zainol Abidin is actively involved in a basic science orthopedic related research and is the head of Tissue Engineering Research Group, which is a part of the National Orthopaedic Center for Research and Learning, NOSRL, in Malaysia. His research interests range from biomechanics to cell biology and fundamental research involving cartilage biology and stem cells applications. He has published over 100 peer-reviewed articles in many peer-reviewed journals and supervises many postdoctoral, doctoral, and other postgraduate as well as undergraduate students. Without further ado, everyone, please join me in welcoming Professor Dr. Tunku Kamarul Zaman, Tunku Zainol Abidin. Prof, virtual floor is yours.
Okay, thank you very much, Dr. Amit. Thank you for such grand and welcoming opening speech. Thank you so much, everybody, for this for allowing me to talk for the next half an hour. For that, I need to apologize up front because I'm going to run through quite the slides because I thought it was longer than that. But today, my topic, I'm sure you've seen here, is on orthopedic implants. And being an orthopedic surgeon, I thought this would be a nice way for me to talk to the audience. In particularly about the biomaterials as well as the biomechanical design aspects and how that has changed over the years. Okay, so basically what are orthopedic implants? Orthopedic implants is a medical device which surgically placed into the body to restore function. And whilst this implant is meant to restore the function, it also replaces the function in many ways. You need to understand that as you grow older and there's trauma and damage, there's always something happening to your body due to accidents or some sort of damage towards the tissue, and you need replacements. And that's what orthopedic implants are for. Interestingly, I went to try to go and Google it up. I found in Wikipedia, and they kept saying that the orthopedic implant is to replace a missing joint of bone for damaged bone. This is not true, okay? Because orthopedics are not just bones. For whatever reasons, this ideology that the orthopedic surgeon is a bone doctor, that's not really true. So to be honest, we take care of the limbs, anything from the joints, the soft tissue, the bone, the muscles, and many other aspects as well. So I'm going to start my lecture by talking about orthopedic implants, and here you can see a few of them up in the front here. I wish to elaborate certain implants and try to make you understand why these implants are seen here today. Obviously, there are changes to what you see today, but let's give you a brief tour of what happened in the past to understand our current and how we see for the future. So the first surgeon who actually talked about surgery is Joseph Lister. He is a surgeon by right, but he's famous not because of surgery, but rather for the antiseptics. And I think all of you know him as the mouthwash called Listerine. which they use it from Joseph Lister, and he was a surgeon. But he wasn't really into implant, but he introduced the importance of having to sterilize implants prior to its use. But the first reported history of implant use was Sir William Abbottot Lane. And in fact, I only knew that very recently because he is not famous for the first person to use implant, but the place that he did it with the orthopedic nurse who later became a doctor, James Agnes Hunt, was very famous at that time in Wales. Okay, and the man that would be made the implant famous was this chap called Sir Robert Jones. Okay, so, and you can see the picture on the right there is Sir Robert Jones with Agnes Hunt, and they had a hospital name after that. I'm taking a moment just to write down there a friend of mine who passed away about a few years ago, a good friend of mine, James Richardson, and he used to be the professor chair who resided in Oswestry, where the hospital was located. So this was the famous hospital where implants began to submerge. Now, the first implants they came out was made of steel, and they found that corrosion became a problem. So they, instead of just using pure iron, what they did was they used a mixture of vanadium iron. Okay, it's because it's supposed to be stamina and elasticity in the material. Now, this was the first time that humans encountered looking at the designing of an implant. And one of the first aspects they encountered was not just the design of the shape and how it contoured towards the body, but rather of the implant itself. And because at that time, iron and vanadium had problems and delivered a lot of toxicity, this became the first joint contentious issue that made them think of how to make things better with implant designs. So they came up with another material called Cobalt Chromium, and at that time they had a mixture of it called stellite and Vitalium. Now the thing is, these things were very good. They're very hardy. They're very strong. They were even stronger than steel. The problem is this material were quite expensive and difficult to mold, as you can see in one of the implants where they use 316 stainless steel against the satellite overlay of cobalt chromium. These became a big problem and was limited in its use over the time in 1930s. All right. So they look at another material. The next evolution was molybdenum. As you can see, that molybdenum is a very hardy material. It doesn't react. The only problem is trying to do this, combining molybdenum with material, they don't react very well. And only in the past about 20, 30 years, we've seen a real composition of a strong material when they had a good combination due to better forging techniques of steel. All right. But in the end, what happens after the post-war in 1940, the stainless steel era for orthopedics started to reign. They became the biggest supporter of orthopedic usage. And one of the famous surgeons was this guy, Martin Kirschner. Obviously, he worked during the 1940s. You can understand this was the time where Hitler was heading Germany. But having said this, he came one of the Germans who came up with a lot of designs, a lot of material, and he came up with what we call as a Kirschner wire. All right, and this became one of the hallmarks of orthopedics, not using all the plates, but rather these implants which fix bones very quickly. And one of the things that the British became envied was at that time all their pilots, when they were shot down, they were kept in hospitals on a stretcher, and they've got traction to just hold the bones, and it took them something like 3 months to heal, whereas with wires and everything was holding the fracture very quickly, and these guys were in and about in less than 4 weeks to 6 weeks, and they were flying again. So they had a faster chance of training pilots in Germany. So he developed this material and the technique that used even today. I still use what we call the K wire. All right. So later on, from that wire, they decided to put into trauma implants, being Kirschner being the first guy who talked about it. And they've looked at usually last time they used a carpentry screw into patients. Obviously, a lot of patients died of metal toxicity at that time. But later they converted to using the same material, stainless steel, introduced by Kirschner, and later all of this became very, very common. And these are the modern kind of screws. They've come with all kinds of designs later on. They had threaded, non-threaded screws, and they had like this, which is slotted nails. You can see here, sorry. You can see, let me just show you. You can see here, the slots here so they can put the wire in, guide the screw, and the screw can go towards the fracture site and repair them. So after screws, they figured out, oh, why not we came up with plates? So the first chapter talks about plates like these to fix fractures. So screws were nice if you can put it against the bone to buttress this, but if you don't have enough bone to hold it, they come up with a plate that puts screws in there so that the whole fracture becomes reduced, as you can see here. So this is a new method that started to came about, and these were later introduced only after the war, not during the war, okay, during the World War II, okay. So later on, remember we talked about Kirshner. Kirshner also helped to develop one of these things, and these guys made it bigger. So imagine a small wire, this thin. Now you make a wire this big. So that thing now comes up today with a nail. The nail today is still being used as the hallmark for long bones, for example, femur and tibia fracture, including but not the humerus, not the upper limbs, but the lower limb, because they're stronger, they're rounder. They don't need to strip a lot of soft tissue. It's easier to come in. And one of the things that they have is they have a load sharing effect rather than the load bearing effect of the plate. Hence, what it means is now the bones even heal even faster, and you can even walk on this on the earlier part rather than the plate, where the plate can ultimately break if you don't load it, you overload it, so to speak. So later on, these guys, the Rush brothers from Rochester, Minnesota, which later became part of the famous Mayo Clinic. They developed the techniques later on, which is the intermediary rush nails, and you can see that those nails, they're not really the nails, but actually the bigger form of the wires made by Kirshner. But later on, another man called Kirshner, in German, Gerhard Kirshner, came up with something called a slotted nail, a K-nail. And this was used for many, many years. In fact, one of the prerequisites during my time for training is that if you're not able to put a Kirshner nail for somebody with fracture, you cannot graduate as an orthopedic surgeon. But today, I think these nails have long disappeared in many of the hospitals. Probably in the KKM, they still use it because it's very, very cheap. And it doesn't require any image intensifier like what we use. I mean, doing an X-ray during the operation, you just open up the wound, put the nail in, and slot it against the isthmus to hold the fracture site, and the whole thing, voila, the nail is good enough for you to walk on, just like any interlocking nails. Later on, there's something called an external fixation. As you can see here, these are the external fixation. This was recorded by this guy called Clayton Parkhill, but the guy who made it famous was this guy called Elizarov. So if you heard, oh, I'm putting on Elizarov is actually referring to him. He's Russian. The first Elizarov he made, he borrowed those wires and wheels from a bicycle shop and started putting on his patients, but he became famous after that. And this area now in Kurgan is known as the Elizarov Institute. Unfortunately, he died in 92. So a lot of his work is still continuous. And at that time, they did a lot of funny things. As you can see here, he is a chain smoker. While operating, he couldn't take it. He started smoking as well. And you can see even his patients started smoking. So everybody was smoking in Russia. So for whatever reason, it was a miracle even with smoking that the fracture actually healed. Okay? So this is some of the historical background. Then we talk about later artificial joints. Now, this guy, Sir Stone Jantley, he is a very famous chap who has invented what you know today as a hip joint replacement. And his success was not just using stainless steel, but then he adopted the use of Cobalt Chromium, okay, to make the implant better. And if you look into this design that's done by Charlie, the design has not changed much since it's introduced in the 1950s. So this was his contribution to the end. Total knee was something else. Charlie was famous for the hip, but his material was later being used with the knee. Frank Guston was one of the people who designed this material, but he used the same concept of stainless steel with cobalt chromium. But as you can see, the design is very different when you have today, and he had the independent use of these joints, which today I'll show you later is a lot more advanced in the way it's done, but the principles remain the same. So let's go back. What happens after that? So I was talking about post-war area. I've spoken about the plate, the screws, the nails, the pins, and of course a combination of design to exact the screw. So if you can see this is like a normal picture of a fracture of the ulnar radius of your arm, not really arm, the forearm. Okay, and you can see that there's pins and holes, and these were kind of common in the 1950s and 60s. The difference is, I know this picture is not 1950s, 60s because the designs are different. The plates are not like these. You see, as you can see, those are areas where you have openings. There's not holes, but rather designs of the plate to make sure that the plates have certain areas which are exposed and not touching the bone. So they call it low contact. So this low contact was only designed in the 70s. So these low contact compression plates were later put in, but it was kind of common to see this kind of plates even back in the 50s and 60s. Right. So let's look back in 1960s in implants, and these were the kind of implants that you can see. So during the development, when I said the plates to come in, not only the plates and trauma sets came in, like the ones you see here, these are all called plates, and they've got different plates for different parts of the body. But later on, during that time, until the year 2000, you see, as all these implants started to come in, and I said just now, the hip now become a lot more stronger, and not just combination of stainless steel and cobalt chromium, but they've now added on titanium, which they felt was stronger and had a Young's modulus which is closer to the bone, and therefore much preferred. They talk about fusion implants. They've talked about knee replacements. As you can see, these are knee replacements. They talk about SI joint implants. And the other one they talk about at the back is all of these spinal implants, which then became the big thing, especially in the 70s, 80s. The development went very quickly, and from the 80s came up with the arthroscope, and then they became laparoscopic. So now they've done the technique. I remember in the late 1990s and early 2000s, they were putting scopes at the back to go and put screws for the spine, so you don't have to open up your spine in a very big fashion anymore. Okay, so as I mentioned just now, improvement in blade designs. The origin designs were just like this one plate. Later on, they created all these holes to reduce the weight of the plate, but also to reduce the contact to the bone, because the bone can only grow if they have a good periosteal or blood supply. So if the plates were too much in contact, it lose some of the bone contact and creates the pressure, so they reduce the pressure by creating all these areas. Further designs later on in the year 1960-2000 was to make these holes slotted, okay, and longer. The reason is they can then screw it out nicely and move the plate to get rid of the best breach and then lock it inside the plates. Later on, stainless steel low carbon type L316 stainless steel, which is standard even for industry standards today for plates in orthopedics. Later move on to high titanium alloy implants like these ones. And we know in orthopedics that this is a titanium screw because the colors are different. And what they've done in the design of these titanium plates is introduced. As you can see here, there are threads. You see this? These are threads. And these threads held the plates down and hold it in a very locked position. And with that locked position gives you stability. And not only does it give stability, you don't even have to make sure the plate now touches the bone. It can sit above the bone, and therefore you have better blood supply. So these were improvements, small improvements we were seeing in the 1960-2000 era. And of course the techniques change, as I mentioned before. You see, like these, now they've come up with a new technique when they thought in the early days, that you can now put that slotted hole in order to go and create, you can do an offset, and what you do is you get a compression outcome, and therefore compression will result in better healing. You can also contour the plate and try to rechange the configuration so that you better purchase. Okay, these were techniques developed by the Swiss group, the AO Davos group, and they've got institute there who talks about all of these things and provide training for around the world. So these techniques was very interesting to see the evolution of not just the implants, but rather the techniques themselves. Okay, so the implants change because the techniques change as well. Not the design change was not just because the biomaterial, but because it changed with the technique. So later on, during those days, the implants became better. Remember, we talk about just those knee implants done by Guston. They only had two separate metals. Now it's not. It's actually one piece metal for the femur, one piece metal for the tibia. And this became a standard practice since 1975. This was patented, and today the design were all based on this. This design here, you talk about the hip, was originally Charlie's just a ball with a big stem that goes into the femoral region. Now it changes. Now it comes up with a femoral head. It has a liner and it has a shell. We also saw joint replacements. This was somewhere at the end of the 1990s, even the early 2000s. They were talking about disc replacement. This was a misnomer. They don't do it in lumbar. Usually they do it in the cervical region. All right, this one is a misnomer, but it's not really that popular because its failure risk is high because they're very new. Similarly, the ankle was also the one that became fashionable, but the results were not so good. The one that lasted a bit longer were the shoulder replacements, and the elbow. Okay, not as good as the hip and knees, unfortunately, but this lasted quite much better than the one that's really done for the ankle, which takes up a lot of load, okay, as you walk. So later on, not even later on, in fact, this was later produced by Chandy in the 1960s. They talk about cement. So this is one of the implants that people tend to forget. They always say that implants talk about metal. Okay, they talk about the design of the metal implant, but really in this case, they came out something called the bone cement or polymetal metacrillate, PMMA. And what it does is this picture here shows that you can mix a monomer against a polymer of methacrylate. And what happens is at a certain time, this exothermic process will occur around 9 minutes. And what you have is a hardened bone cement, which if you put inside the slotted of the femur canal, you put in the stem, it will harden and hold the implant. Later designs in the late 90s, 1990s and early 2000s, they talk about cemented and uncemented. The difference is they don't use cement anymore. What they done is they were able to coat the material with hydroxy apatite, and therefore they don't need the cement anymore. So these were advances seen during the early 1990s to 2000. Another one that we've seen also is, other than the trauma set of metal, they also came up with non-metal implants. And this is an example which I've used before. Silastic or silicone joint implants like these for a patient with rheumatoid arthritis very badly. They cannot use the hands anymore because they couldn't move. And this just replaced into the joints, as you can see here. Last time, when the times of Gunston, they used metal on metal, okay, because they were rubbing and the failure rate was high. Later on, for knees as well as hip, the design came with something called ultra high molecular weight polyethylene, which was again produced by even the time from Chanli. They were not so good because the manufacturing process was not improved, but over the years the improvement was so good that these became very strong. However, I show you later, even with that, they still need improvement because the outcome for this showed that the lasting because of wear and tear, it only lasts a good 15 years, where you probably need another surgery to replace the inserts. All right. We saw later on that with the polyethylene, with the silicone, they came up with anchor sutures. In surgery, we need these anchor sutures to pull in soft tissue, not the bony fractures. That's why I said the definition made by in Wikipedia is very wrong, to give the impression that orthopedics just talk about fixation of bones, the implants that used for fixation bones. As you can see here, we use anchor sutures which then pull the soft tissue in repair. This is a job, a work that I used for shoulder, for example, that we use try to repair the damaged rotator cuff tissues in the shoulder. All right. So we also seen metals not being very hard. It used to be wanted to have the K nail. We wanted to have interlocking nails, which are very strong because you need to put in the body weight. But some metals don't require that. And what happens with children when you have more flexible bones? So they came up with flexible implants like the ones you can see here. These flexible implants also came up with the biodegradable implants, which can use it for anchor sutures. So with all of this happening and all of this what's happening in the future, let's look at what will happen in the next few years. The orthopedic implant is a very promising market. As you can see that over the years, the use of ceramics, polymers, composites, cements, all that have been increased. And you can see that there's going to be a top trend up to the next 5 or 6 years. And God knows, because with the number of people ever increasing, with the number of people aging, this is expected to grow exponentially. In fact, I've taken this data based on the market survey on some of the top market surveys looking at what the implant market is going to be like. As you can see, this market survey is getting bigger. And especially Asia, with China becoming more and more affluent over time, this market is going to be glut. And we expect by 2030, this segment now will overtake even Europe size and somewhere close towards the North American market. So it is a huge market, and it's something that's growing over time. Euro market, again, I'm just going to show you that the good thing about the Europeans is that they got a very good total hip arthroplasty, and because they recognize the fact that Europeans are going to be an aging population as a whole, and you can see that number of procedures are just staggeringly going upwards in a very good trend. So that means that there will be more implants in use, and there are more demands over the next few years. Asian market, as I mentioned before, look at the China orthopedic. This is shoulder replacement implant market. It's almost as if the growth is exponential, and you can expect it to grow even higher for the next at least another 20 more years. So we can understand that will also grow over time. All right. So what is it now today? Now that we are coming up to the stage of between the year 2000 to 2030, what will we be looking at? Stainless steel is still being used today. Even though there's titanium plate stainless steel being used, because now they make it very, very cheap. And it's produced not just all the big companies anymore. In fact, they don't produce by the big companies like the main players like AO and Striker, but rather even small companies now, tech companies in China, they're mushrooming everywhere. I go to all these orthopedic conferences. There's so many companies that are selling this stuff, and almost everybody is selling, and there's a glut for it. Okay, and they're selling things very cheap. I remember a plate last time used to cost about 15 US dollars. Today you can get it in less than 4 ringgit. Okay, so imagine what the market has done. So the 316L is still a very strong market, especially for countries which are developing. Okay, and whilst that's having said, there's also a glut because a lot of countries now becoming from the underdeveloped or developing into developed nations, and their countries are becoming rich. Therefore, the use of cobalt chromium, titanium, ceramics is another one, the new ceramics, because it's supposed to be harder than steel, but a lot more brittle. And the use of new materials like PLLA and tantalum are new stuff that's coming into the market today, and it's now being used regularly as compared to what it was used before the turn of century, okay, before year 2000. Okay, so I'm going to just mention this. In fact, I'm just going to go through since I'm running out of time here. The 316L is very good. All right, it has shown a good track record. The only problem is it's very strong, and the problem is it creates a sort of stress shielding effect, which I'll try to explain to you in my slides on biomechanics later. Titanium is a lot better, but the pricing is the issue, so to speak. And titanium contains the titanium, aluminum, and vanadium, and today is still considered the best material in terms of strength as well as its young modulus properties. All right. Cobalt chromium, as I mentioned before, again, is also very good. But the problem with cobalt chromium, it has a very high Young's modulus, unlike bone, and the risk of stress shielding is there. The only thing that now cobalt chromium is being used is only for your hips and knees, the joints, because you need to have high resistance. But you will see later, as a surgeon, I get to see a lot of them having what as bone lysis, because the bone then ended up with this stress shielding effect, and they don't grow very well. Right. So here is the standard picture for the stress strain curve. I think most of the audience are engineer, so they understand this very well in any material. But more so, it's slightly different for us, for orthopedics and biologists, because you have a viscoelastic material to consider. So some of the stuff that you see is for implants is more likely to just this and here, but we do see the larger part of this plastic zone, because we deal with implants that are not just metal. But what I'm going to show here is, if this was the cancellous bone, and this would be the cortical bone. Where's the cortical bone? Yeah, it is. All right, cortical bone. Well, let's see here. Okay, it'll be cortical bone here. What happened is, if you talk about these bones, is that if you look into all this titanium as well as stainless steel, these materials have high Young's modulus compared to this, and this is the reason why you have an effect. Whereby if you were to put the stress on the material will take up the load and not the bone. And this goes against Wolff's law, which states that in order for bone to grow, they need to have mechanical stimuli, and that they do not get at the end of the day, and the bone gets resorbed over time. So a best biomaterial for bone healing would be somewhere along this line, and they talk about PEEK. Whereas titanium is closer to this, but PEEK will be the next material in choice for implant replacement. The problem is with PEEK is, just like ceramics, although not as bad as ceramics, which is glass, they undergo quick fractures, and they have they easily undergo fatigue. Okay, so implants application now, we can see a lot more things. And I mentioned before, again, I'm going to mention orthopedic does not equal to bone doctor, so they do other things. So they work on bioresorbable. They use this for ACL reconstructions. Lots of you want to play sports, you end up with ligamentous injury of the knee, especially the ACL. I'm sure everybody heard about the ACL. Probably not many know what ACL stands for, but they know what ACL is. In fact, most of the times we use bioresorbable screw to hold the ligaments when we construct your ACL again. All right. So the non-metal implants are now growing in the markets. We're talking about flexible implant and soft tissue repair, and the areas of my interest will be cartilage, tendon, and ligaments. And these are already, some of them are already available in the markets. Okay. So these were the materials now that you see, other than the ones developed before, and you can see some of them still being sold. The newer generation materials like these, they have antiseptic implants where they've actually put coated implants, not only with HA now, with antiseptics, so that they don't allow any infection to occur. They make into biodegradable implants like these. These are made of cellulose, where you have people where you have delivery, and once the tissue have regenerated under something what we call a script substitution, the material then gets reabsorbed. Okay, the later ones that you can see here, these are pit titanium cages. Now these are used being used at the spine. Okay, so to stop them from getting resorbed. So what happened is these materials now not only are they good at holding the load, but rather of the tissue itself. So these are now being used more dramatically or more encouragingly in the market. The new implants in the future today, these are done in many ways. Implants supporting biologics, and in this case, you can see all these implants happening, being introduced now. Okay, they are extracellular materials that support cell growth, and these are one of the examples that we've actually used in our labs. Okay. So I'm just going to show you one of the materials. I'm going to rush very quickly because I'm running out of time here. So if you understand that cartilage is not bone, okay, please, whatever you say, cartilage is not bone. And when you have a damage on this tissue, if you can compare that damage there to the normal athletic cartilage, you can see that there is a very distinctive difference between a damaged cartilage and that of bone. You can see there's normal cartilage and this is bad cartilage, everything coming out. There's no way you can put a plate inside. There's nothing you can just replace with just some sort of tissue somewhere. It doesn't work. All right. So there are many ways that you can come to. So I'm just going to head up very quickly. So what they do is, all right, okay, let me just show you. So what we've done is an ACI implantation. This one technique is being introduced as being implanted over 1 million patients or more. And you can see the damage there. The bone is actually exposed, but the cartilage, the white color stuff there is all the cartilage. So this is a biomaterial designed specifically for its use. You can cut it out to size to replace that damaged area, and in this case, it's only being used just to cover the damaged site. Okay, the damaged site. So you can see it's very flexible. It's healed up, and in some of the stuff that we do is we put cells on top of that so that they can carry the cells and transplant into the site to help regenerate the cartilage damage, which otherwise cannot be replaced. Okay, and it glues up the site as you can see here. Put it inside, patch it up, deliver the cells inside here, and voila. Okay, and you switch it all on. Right? Okay. So we've done quite a bit from our own experiences or some of our publications, just some of them, and we look at how we can do this for the spine as well as in the cartilage. And we've shown that we were able to improve it now even better, not just with the implant designs, but rather with putting in cells. So the new implants of the future until today are looking at cell therapy, gene exploration, as well as the delivery of growth factors. Okay, so tissue engineering and region medicine is the way forward. And you can see here, this is the vacanti mouse that you can literally grow something in the lab and put it here. This is one of the biomaterials we use, alginate calcium beads. And you can see here after pre-treatment in post-delivery with stem cells as well as growth factors, there's nice healing of the cartilage, right, in our subjects. So which will prevail and make it bigger? There are three main principles in developing marketable orthopedic implants in the world: cost control, production, innovation, and the technology advances. So these things will be in the way forward of what you can see. And as an industry, cost control is the biggest thing. Okay, so if you look into the cost control, whatever you do in your research and design, you have to look at the total cost of the material. As you can see, if you look into this, to be honest, I think the gold implant looks pretty good. It's not hard, it's soft enough, it probably has, if you mix it nicely, that you can have an implant which has a Young's modulus closer to that of the bone. Unfortunately, you can see at the price per gram for gold compared to something that you're using for steel, there's no way gold will be the standard of next implant in the future, unless you can know where we can find big reserves around the world. Okay, so you look at some of the implants that can you bring down towards the pricing over time, and that will be the implants of choice. So cost will determine how the industry shall research and move forward in the near future. So the choice of implants makes a difference because, as you can see, so many publications before talk about the cost effectiveness of an implant based on design as well as its content. All right. And over time, one of the things that you have to look at implant is ensuring that implants don't fail. In this case, you can see stainless steel is now becoming less and less popular, even though how thick it is, because you want to cut down the weight, you make it very thin, and these tend to have a breakage over here. If you make it too strong and too hard, especially in older patients, this just cut out. You can see that? Now it goes right into the hips. So implant failure will be an issue in the near future, right? Even titaniums also fail. So if you're looking at new designs, new way forwards, you can see that they are not as strong as I showed before in the picture. So they tend to break over time, and these are still areas that needs to be explored over the next many years. Poly, remember I told you about poly implants? These are just wear and tear because of the weight issues, and they need to be replaced. And so far, for high stage users, sometimes the poly has to be changed even up to 10 years. So your surgeries have to become a lot more regular, right? Now, I need to take a few moments here. I know I've taken a bit of time here, is to talk about design implants that people tend to rush and go to market. This is a good example of failed implant design, and this market is called the metal on metal implant. It promises a better wear because you don't have the polywear what you saw just now. So they say, oh, it can last longer. But what happens is those patients that came in, they find all this metallosis, this black stuff that comes out, the black goo, and that damages the tissue around it, and you cannot implant anything new. But that's not just the main issue. What happened was patients also end up with cobalt toxicity, leading to renal impairment in a number of patients. So these implants were the worst, and they were marketed without proper clinical trials, and it's allowed under FDA rules. So it's very interesting how it works. So the issues with these implants now, we're not talking about just the implants themselves. The manufacturing process needs to be looked into. The human biological demographic difference and the poor copycats out there. So you need to be very careful, all right, in your designs, okay? So the lack of evidence is another thing. And that's even going into countries like the US, even for today. This was a nice paper looking at the issues here, and I was reading through and talk about the 51K pre-market notification, meaning that this 510K is that you allow an implant to be used even before they are being tested properly in the market, based on the fact that previously there's already a history of using the same implant with some changes here. Okay. All right, so we will talk about this in a while, and I wanted to show you a video of it to try to explain it better than the way that I tried to explain it. This was by John Oliver. Just to give you, okay, I'm now running off a bit of backstage here, so give me a few minutes.
Medical devices.
All right.
Modern day miracles that range from contact lenses to pacemakers that can heart beating to hip replacements that can transform lives, as this woman will tell you in a solemn testimony.
I was very frightened to do a hip replacement. I really didn't want to do a hip replacement.
That's fascinating to see her. She's dancing as if she never got it.
Okay.
Admit it, you didn't see that story ending with a breakdancing group of 50-year-olds called, and this is true, Moms in the Hood. The medical device industry is huge. They constitute a $156 billion market in the US, and more people have one than you might think. About 32 million Americans, that's 1 in 10.
Okay.
Have at least
So
one medical device.
let me forward you to the about how.
Well, you naturally assume that medical devices are extensively tested before they're put on the market. That is not necessarily the case. Even devices that go inside you may never have been subjected to a clinical trial. And don't feel bad if you're suddenly surprised by that. One researcher found that many doctors aren't aware of this either.
We were surprised to see how little surgeons knew in our study about the approval process for devices, that some of the surgeons seemed to think that, well, the FDA had taken care of this, that of course these devices had been tested in humans.
That's terrifying. Even your doctor may not do one.
Okay, so
it wasn't actually a bad idea.
Sorry.
It means you get your own hanging water bottle that you can stuck on from your bed. Most products get cleared. If you're a loophole in the system called the 510K pathway. It wasn't actually a bad idea when they initially came up with it. Basically, the FDA wouldn't make companies go through a strenuous testing process if their device was substantially similar to one that had come before. That way they wouldn't stifle innovation. But it was only supposed to apply narrowly. The problem is nowadays, more than 80% of medical devices are cleared through the 510K loophole, including around 400 implanted medical devices every year. Those are ones that go in
Sorry.
inside your body. And that whole idea of being substantially equivalent to devices that came before has some real problems with it, because it can essentially become a high-stakes game of telephone. Let me show you. In 2008, a company called DePuy received FDA clearance for a hip replacement based on its substantial equivalence to 6 previous devices, all of which were cleared because of their similarity to devices before that, and devices before that, and before that, all the way back to hips that were on the market in 1975. Now, incredibly, some of those devices have since been taken off the market by their manufacturers because of their high failure rates, but under FDA rules, DePuy could still use them as a basis for getting their hips cleared. And the thing is, DePuy's hip really would have benefited from clinical testing in human beings because there was a big problem. While it may have looked similar to its predecessors, it was crucially different in that both its ball and socket were made out of metal. And it turns out that when metal rubs against metal, bad things can happen.
Okay, sorry.
The tagline for a movie about a masturbating robot. Those metal hips began to degrade with wear and tear, and the consequences were grim.
Orthopedic surgeon Dr. Herbert Huddleston of Tarzana testified at trial he's taken out dozens of metal-on-metal hips that failed. In the worst cases,
bone and the soft tissues around the hips started to look like pieces of cheese, except pitch black pieces of cheese. Sometimes we get in there and it looks like it's filled with old oil that you drained from your car.
The DePuy's parent company has since spent more than $3 billion.
Okay, sorry. So I'm only running out of time, so I wouldn't be. But if you like it, I can just send a video with this to the organizers in the case show. But it's a very interesting way. I think he's more articulated than I am, John Oliver. So perhaps he can explain to you that there's a flaw in the system, so that the lay person can understand there are flaws in the designs of the implants themselves. So there is a lot more room for us to make improvements for the near future. Okay, so what does the future look like for orthopedic implants? Again, I'm going to mention here that cost becomes the big thing, and now they're talking about zirconia titanium implants. I've seen that before, but how well it does, whether or not it will cause more problems, is something to be seen. But so far, it has been one of the things that's talked about, right? And of course, now you heard about personalized medicine, where you talk about big data analytics of huge number of population studies, 3D implants this design and implants the patients so that they can work better for them. Novel biomaterial designs. Biomaterial has been classified into three: the first generation, second generation, and third generation. If you guys remember my earlier talk, I said just now, the earlier part of my talk, I did say that in the initial part was to talk about bio-inert materials. You just try different types of materials to make sure that they don't leach and cause problem to the body. Then later on in the year 1960 to 2000, you talk about having to have biodegradable materials, not just metal implants. But now we're moving into third generation, where we can look at stimulating specific responses from molecular level. For example, here you have drug-releasing implants, either for the treatment of infection or to increase the bone repair or cartilage repair. This new way of third implants. And now they're talking about smart implants, where you use engineering in living materials, and biomemory implants, where you can then reprogram them in many ways, including genetic manipulations. Biological active implants are the next stage. Okay, so you talk about implants that can literally react with the body. So this will be the future of what the implants industry design are looking at. Okay, they may be more costlier, but if you produce it in large numbers, the cost will then drop. So these are the way forward from where I can see. In our own experience, we've already looked at biomaterials plus another sort of enhancement. In this case, we use growth factors, BB, in encapsulation, but we use it in nanoparticles. So you're talking about now moving from the second generation to the third generation. Similarly, we use the same sort of concepts in our allogeneic systems in the transplant of cartilage. As you can see here, the PDA chitosan delivery mechanism actually works. So this will be the near future of the implants design. Okay, so I'm going to finish my lecture here talking about this implant. So I need to remind the audience that the development of orthopedic device is a very promising and exciting field. Okay, and we've seen how it changed over time. And while you talk about the old technology, you need to be sure of the latest technology that comes into the market, and it's huge, potentially huge. But you also must understand there are its whereabouts and failures in it. And therefore, perhaps these are things that you can work towards developing over the next few years in your research. I'd like to thank you for the time to listen. I'd like to take time to thank all these people who've worked in my lab to produce all the results that you see. I'm just taking a moment just to put in memory, because while looking at the history of all the named surgeons, this was my trainer who's actually in the University of Liverpool, which is one of the famous centers for doing orthopedic research. And he actually taught me that some of the stuff that you've done here needs to be sure that you don't end up in patients and killing them. Thank you so much, and apologize for taking a bit more time than I should have.
Thank you so much, Professor Tunku Kamarul. It's really exciting and inspiring. Exactly. All right, so we have a lot of question here. I don't know whether I have a time or not with it, because we have a keynote session after this. Well, I will allow maybe one or two questions I will read on the comment. Okay, so the first one I will read. Currently, is there any implant designed and fabricated by Malaysian? And can
Okay,
Malaysia become the hub in producing medical implant?
okay. Number one, is there companies actually producing these implants? Yes, there are implants company. Apa nama company? I forgot the name of the company. It was in fact they were producing it and supplying for the KKM hospitals. Okay, there was a company that does that. The question now is, number one, why would we want to have a Malaysian company to go and deliver our market? Okay, number one, it has to be cheaper than the parent market, correct? Okay. Number
Yes.
two is you would like the implant to be better than as a market, what we have. The problem with those implants that were supplied to our local market was that they are actually cost more. So it defeats the purpose of having an implant, and it was no better for the implants that you can get probably from China, India, or Turkey. Okay, that was the issue. So if Malaysia wants to be a hub, it needs to work on two things. Either we can cut down on costs, or we can cut down on we can make into better designs that no other countries can do. So in order for this sort of implants to be well established, well marketed, and well accepted, you need to work again. I'm going to mention here, talk about cost, talk about innovation in designs, okay, that nobody else can do. If you're talking about cost, there's no way for you to go and compete the Vietnamese or Chinese market. They can have costs way down to levels that you cannot imagine. Okay, you can do it in India, imagine the cost will be so cheap. So cost is not something that we can compete. But if you're talking about implants that's very, very genuine, very, very market stabilized, very, very specific, this is the way forward. Okay, so that's my opinion for that.
Okay, thank you, bro. I think that's all for
Okay.
the Q&A session. I'm really sorry for all the participants. There are a
Right.
lot of comments here. A very interesting talk. Okay, and eye-opening about the implant and so on. So I'm really sorry about it, because we are running out of time. Okay, and then we are grateful for the time and effort for you, Prof. With your busy schedule to share your thought and deep knowledge with us today. And we thank for your valuable contribution for this conference, Prof.
Okay.
Okay,
Thank you.
well respected delegates, that wraps the keynote session, and we don't stop there. And for participant, iMediTech conference, okay, we will continue with the second parallel session at 2:50, okay, 2:50 p.m. Hence, you can access the meeting. You can access the meeting room via the WebEx link provided in the email sent to the registered participant of the conference. Should you encounter any technical issue, you kindly requested to communicate with the committee. And meanwhile, for the ICBME participants, you may please stay tuned here. Okay, with that, I would like to say thank you. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh and greetings to all. Welcome to the International Joint Conferences on Medical Engineering, Science and Technology, IMEDIS 2021. I am J. Suman Puspanathan from the School of Biomedical Engineering and Health Sciences, Faculty of Engineering, University of Technology, Malaysia. IMEDIS is a joint conference between International Conference on Medical Devices and Technology, IMEDITEC 2021, and International Conference on Biosciences and Medical Engineering, ICBME 2021. So without further, before we proceed further, ladies and gentlemen, please go ahead and give a like and subscribe to our YouTube channel and also the Facebook that you are watching live right now. So today, ladies and gentlemen, we are privileged to have our distinguished speaker as our first invited speaker, Professor Dr. Engineer Habil Jenshausen. Professor Jens Hausen since 2005. Yeah. All right. Okay. Sorry, there are some technical issues over there. Professor Jens, just to double check with you if you can hear my voice.
Yes, now I can hear you. There was some transmission issue, but now it's working again.
Yeah, looks fine now. All right, we shall proceed, ladies and gentlemen. Sorry for just now. Jens Hausen is a full professor since 2005, and he is the chair of the Institute of Biomedical Engineering and Informatics at the Technical University of Ilmenau, Germany. Yep. He has a master's degree and PhD in the field of electrical engineering from the Technical University of Ilmenau, Germany. He is an adjunct professor at the Biomagnetic Center, Department of Neurology at Jena University Hospital, Germany. Professor Jens is a member of editorial board in several scientific fields such as technology. His research interests covers the field of bioelectric and biomagnetic, including and functional and many. Ladies and gentlemen,
Looks like we're facing some technical problems again. Yeah, the line is slow. There's a comment in the chat that the line is slow indeed. Now you're
So
back
sorry
again.
for that. Yeah, Prof Jens, I'm sorry for the technical problem. Well, I'm not sure where did I cut off just now while I was reading your biography. However,
we were pretty much true with the biography.
okay. Well, okay, without further ado, let's invite Professor Jens Hausen for his presentation on dry electrodes for electroencephalography. Over to you, Professor.
Wonderful. Thank you very much, Zaman. It's a pleasure to be here, and thank you very much for the nice introduction. And technology is good, but not always that good. So we still have some telecommunication issues to solve, obviously. Can we put on my slides, please, so that I can have the screen shared? So there should be play your first slide while I do.
Yeah.
Share it. Okay. Share screen. Okay, there we go. Yeah, perfect. Wonderful. Thank you very much.
Okay.
Sorry.
Great.
Yeah, so thank you very much for the introduction. And now let's speak a bit about medical technology. And I would like to go and address with you a little bit the topic of electroencephalography. Electroencephalography is the measurement of electric potential differences on the head with at least 2 electrodes, as you can see here on the slide, but with, in most cases, actually many more than just 2 electrodes. This technology of measuring potential differences goes actually back to the person here on the right side. His name is Hans Berger. And he was the person who discovered the electroencephalogram with the equipment you can see here on the left side. And back then, which was about 100 years ago, actually, in the early 1920s, he was using electrodes for measuring, which looked like this here. And those electrodes are actually needle electrodes. And typically, he would stick those needle electrodes into the skin of his volunteers. This is something which we typically don't do nowadays because our volunteers don't tolerate this so much anymore. Back then, it was obviously a requirement just for the recording technique, but recording got better. And in this small video here, you see how a typical recording in the EEG lab is done nowadays. So you see here my PhD student, but normally lab techs are doing this. So the electrode cap is placed and then the gel or paste is filled into each electrode, as you can see here. And this is what we call wet electrodes. Wet because each of the electrode is filled with a wet gel or paste. This can take quite a while because it requires some skills. And if you have new students, then they easily take an hour for this amount of electrodes. But if you even have more electrodes, then it's even worse. So it might be even for professionals up to 1 hour. And what you also recognize is that we are here in a typical laboratory setting, and it is the same all over the world. When you do an EEG recording, nowadays you are in a lab or in a clinic. So those are things which you might want to overcome because you might want to measure also outside. Then there is an issue with the wet electrodes, which you can observe now. Once you take off the electrode cap or the electrodes as such, then you see that the volunteer or PhD student here is not really looking too happy, is she. And the issue is, of course, something where we would like to improve. And what we would like to do for this is what you see here, on the next slide with actually one of our first prototypes of dry electrodes. So the first thing to observe here is we're not in the lab anymore. We are in the living room of our PhD student, actually. And the next thing to observe is she is all by herself. So she's not getting any help from someone to place the cap and to start recording. So it's just placing the cap, connecting the device, and then start recording and doing a medical grade EEG. And this, of course, opens up the possibility for many new fields for EEG, just like telemedical applications, so that people in one site do the actual recordings, even say in nursing homes or even in other places like the patient home as such. And then at a central place, you actually analyze the data. Now, finally, you can observe one more thing. Once you're done, you take off the cap, place it back to the holding, and then you're done, you go away. You don't wash hairs and you don't have any aspects around the gel or paste, which you typically have with wet electrodes. Now, since one of the topics of this conference is also about sustainability, I was calculating here for this conference a little bit how much this would mean in terms of the waste produced by the EEG. So actually, each electrode has only about 1 milliliter of electrode gel or paste. But you have not just one hospitals. Only in Germany, we have about 2,000 hospitals. So we calculate 200 working days for these hospitals. We calculate 5 patients per day, which is, say, a rough number, just an estimate. So which means 2 million EEGs per year. Now, it is not just one electrode. It is typically like 24 electrodes, which you can use, which you can see on average. So that produces 48 tons of paste waste per year, which is not easy to actually address because it's chloride included. Then when you think about washing the hair afterwards, which is needed anyway, so heating up the 5 liters of warm water per person, you end up with, as you can see down here, with about the amount of 230 megawatt hours of energy. So, and there are some other aspects. So from the point of view of sustainability, conventional wet EEG would be actually quite good to be replaced. And this, as I said, these numbers are just for Germany and not for any other country. So with this motivation, I would like to go into the development of dry electrodes. This is what we have been doing for the past 10 years or so. And the first thing what you want to address is, of course, requirements. So you need to be good in your applicability of your new electrode technology. You need to be good in your reliability. And last but not least, in the producibility. And that has several items like biocompatibility, etc., which you are seeing listed here, and which I will address in the remaining part of the talk. So the first thing is with your electrode, the standard electrode, which you see here on the right side, the standard gold or silver electrode, silver chloride electrode, that is something where you need paste to get through the hair layer. However, if you want to have a dry electrode, then you need to be in a sort of fashion going to the hair layer that you can contact the skin without the gel. That means you need to have a pin shape like the ones you see here. Actually, this one is also only about 1.5 millimeter in diameter, so it's enlarged here because it's a single pin. And these pins, they have different shapes, as you can see here, and different sizes. And this is something which we tested and characterized. And what you can see up here is that we used quite different materials: titanium, titanium nitride, polyurethane coated with titanium nitride, gold pins, polyurethane coated with silver chloride. So they're quite different in terms of materials, and that means you need to go and also characterize them. And this is what I want to show you here in this slide. So we do electrochemical characterization, and I show you for the next 2 slides, or 4 slides actually, just the first 2, but the rest, which I don't show here, they are also part of the electrode characterization and need to be addressed. So impedance and open circuit potential are important ones, and therefore I've picked those 2 here. And the first thing I would like to address is the impedance measurements. How do you do this? You place your electrodes into a bath of electrolyte, typically natrium chloride solution, and then you apply a impedance measurement in galvanostatic or potentiostatic measurement setup. And this works actually for a 2-port or for a 4-port setup. Both options are there. And you typically do this with an impedance analyzer device. We use the HP device for this. And on the next slide, I would like to show you the results for our different electrodes. So you see up here the different electrode types, and you see they are color-coded here in this legend. And what you can observe is, of course, that the absolute value of the impedance is lowest for our gold standard electrodes, so the silver chloride electrodes, the classic ones basically. But what you can also observe is that for the silver coated polyurethane electrodes, we have also relatively low value. And more importantly, the standard deviation, so standard deviation means always the dotted line, is relatively low compared to all the other types of electrodes, which we see here. The worst one is actually the gold pin electrode. When we look at the phase shift which we observed, then again, of course, our gold standard has the lowest phase shift and also the lowest standard deviation. Again, followed by the silver chloride electrode based on polyurethane as a substrate material. And again, you observe here for both the titanium as well as for the gold pins quite significant standard deviations. So this already gives us a hint that the gold pins might not be the best choice, but we'll have a look also at the open circuit potential. So how is this measured? You have a reference electrode, which is a standard electrode, as you can see here. And you have your test electrode. And again, they're located in a bath. But this time you just measure with the multimeter the potential which actually occurs once you immerse the electrodes into the bath. So with this, we have a look at this potential. And here, most of the traces are actually quite straight over about 150 minutes. That means they're quite stable, except for 2, namely the gold pin electrode. Also here, watch this large part of the standard deviation. And in addition, the polyurethane-based titanium nitrate electrode, which have an also not very stable behavior over time. So in this, from this we concluded that our polyurethane and silver coating is a suitable approach for our recordings. Now the next question which we had was, how many pins do we need? So what do we do here? We take one pin, we take a number of these, and then step by step connect them. So basically start with one pin, add another pin, etc. And what you get here is this impedance value, but consider those impedance values are now in kilo ohm. And we're talking about the impedance at the skin. So this is measured actually at the volunteer's skin and not in the bath anymore. So what you see here, the silver chloride gold standard electrode is the one down here with the lowest values. That's expected. But if you have about 10 pins, you're getting close to that already. So in other words, we concluded from this experiment here that roughly about 10 pins, maybe a bit more than 10 pins, are sufficient for our typical recording setups with this type of pin electrode. And this led us actually to the design, which you see here, where you have pins integrated on one base plate, so they're connected all together, such that you have basically one and the same base plate here for so one electrode and not one pin, basically. Now, this is then, of course, put into practice, coded, and then used for measurements. And there we arrive at what you have seen in the video earlier, namely this situation right here, where we have this cap and where we can actually go and use this cap in studies. And this is what I want to show you now in the next few slides. So we compared this on 20 volunteers with a standard wet cap. So we use this dry new setup, which you can see here on the right side, and compared to this to a standard wet cap, which you have seen in the wet cap video. Now what comes out is the following. So we have here, for example, the alpha activity. The alpha activity is the activity which you measure typically over the occipital cortex, and it is related to a relaxed and eye-closed state. It is the strongest activity actually in the brain in the ongoing EEG. And what you can see here is the power spectral density for the dry in orange and the wet in blue. And what you can see is that we have here a nice alignment of the peaks. And this is already the grand average of all the 20 participants. And you also see that the standard deviations are nicely aligning. So in terms of topographies, so we look from the top onto the head, ears, left and right, nose in the front, and you see that we have here very nice activity over the back part of the head. And this is actually the same for both wet and dry. Now with this, we had, with this first measurement test here, which I show you here, we had some issues with contact pressure, because not in all subjects we were observing a perfect contact pressure. And this made us having a look at contact pressure and comfort, because also some subjects complained those pins are a bit too hard. I don't feel comfortable with this. So especially in the temporal positions, people were complaining, sometimes also in the frontal positions. So we have chosen to actually measure the pressure and see how the pressure and the contact impedance actually are related. And this is what you can see here. So you see one of our volunteers laying on the back and having this device, this force measurement device here on the front part of the head, and there is an electrode in here. And then we adjust the force basically and see how the comfort is behaving. And this is what you see on the right scale. There is a discomfort scale. So 1 means I don't feel nothing. 10 means this is maximum pain, take it off as soon as possible. And what you can see here, if you increase the force from just placing it to 4 Newton, then you see that we have an increase in the discomfort basically. But consider we're still in the lower range of the scale up to 10. On the other side, when you see the impedance, this impedance is of course dropping as you increase the force, and that is also expected. And what you can see is that already with 2 or 3 Newton force, it is not very difficult to reach good impedance values anymore. So in other words, the conclusion here is that about 2 to 3 Newton are sufficient for the pressurizing of this electrode. With this, we went on and designed also new electrodes, what you can see here. And what you recognize is, first of all, there are many more electrodes. This is a 256 channel cap. So in the front part, we opted for electrodes which are not having these pins because you don't need this. And then we opted in the second round for smaller pins because typically at the beginning of the hair, well, not in my head, but in most heads, then you have this line where the hair is not very thick. So you go along with those smaller pins, and then you go with the larger pins. You see the 3 types of pins here on the right side. And again, with this, we went and did a study on 20 subjects. And this time I show you results for the experiments which we did with visual evoked potentials. So visual evoked potentials means that you look at the screen and you see a checkerboard where the black and whites are switching on and off, basically black and white. With this checkerboard, which is called also pattern reversal, you have a certain activity evoked in the brain. And this activity is recorded with the 256 channels. And this is what you can see here in the time traces. So you see 256 channels basically here for the conventional wet and here for the multi-pin dry. And again, we have a look at a certain point in time, actually the N75, this is this first peak here. And we see the difference between wet and dry recordings, which is basically not visible. And the same here for this main peak, basically, where you see the difference between wet and dry. And there is basically, again, this is wet left and dry right. There is nothing much visible here. So from this, we would like to conclude that we indeed are able to generate comparable signal quality for wet and dry EEG recordings. We have done now more than 100 subjects on this in our lab, and the electrodes have been used also in other labs, and we can clearly say that the quality of the dry recordings is comparable to the wet ones. The multi-pin electrodes are really able to pass through the hair layer. This is important to get contact. And with our optimizations, we have been able to improve patient situation in terms of comfort, although we are not yet fully happy with that. And well, one other thing which I mentioned already is that we are, of course, able to do recordings without any additional support from trained technicians or similar. So I think I'm pretty much at the end of the time. I still have a few slides, but let me switch over to the final summary slide. So we have this is the summary slide, sorry. So what we have done is over the past 10 years of development and research in this area, we came up with electrodes which are biocompatible, which are electrochemically stable, which have good signal quality comparable to wet electrodes, which are really rapid and reproducible in application. They go to the hair layer pretty easy, actually. We have some issues where we are currently doing research on the contact stability. That is especially true when you do EEG in sports science, where you have people moving a lot. and where you want to record also besides the movement artifacts you have. There is still some work to be done in patient comfort that is ongoing, and we're still a bit too high in costs. That is, of course, something which we're working on, but it's in terms of outlook, one of the outlook points actually. So what we will do in future is we will improve wearing comfort. This is something which is already addressed at the moment. We will do combined usage with near-infrared spectroscopy and functional MRI. And we will also have some modular and adaptable caps for different head shapes, also for babies, for example, and things like that. Yeah, with this, I would like to thank you very much for your attention and thank my co-workers and happy to take questions.
Sanchik, yeah, that was a wonderful presentation, Prof. Jens Hauweisen. We shall now have a short question and answer session, Q&A. Should you have any question, please post it at the comment section right below this video, either from Facebook or from YouTube. And we will take up some of the questions. Yeah, we have one from Dr. Aisha Shafi. There you go, Prof. Jens. Question for Prof.
Yes.
What is your overview on textile-based wearable electrode?
Yes, textile wearable electrodes. Well, I actually had a slide on this one. So that is one of the slides I skipped, unfortunately. So textile wearable electrodes are something which is, I think, also a way to go, not so much for EEG, but for places on the body where you are hairless, more or less. The other thing is when you want to use it for stimulation, then you could use wet textile electrodes. And this is actually what we have done. So we have, for example, used larger patch electrodes with a sponge put into the textile. The sponge is then wetted. So the sponge is only very thin, like 2-3 millimeters. It is wetted with natrium chloride solution, and then it wets both the electrode, which is underneath, and the way to the hair to the skin. And there you can do electric stimulation, like transcranial direct current or transcranial alternating current stimulation. And this is something which we have developed this together with a company, and I think the company will sooner or later commercialize this. So the paper was published in 2018 on this, and we're still in the discussion with the company about commercializing. But wow. I think textile electrodes are something which is really having a future, definitely.
All right. Okay. Let's see if we have any more other questions. I have one question. Okay, let's take this question. Professor Jens Hauweisen, how do you see the future of electroencephalography in the near future, looking to the ongoing pandemic situation around the globe? Yeah, how do you see the future of this?
So electroencephalography is like a standard technique, both in research and clinics. In clinics, it's mostly used in neurology for diagnosis of epilepsy, for diagnosis of other diseases. There it will not change also in the pandemic situation, because no matter what the pandemic situation will be, people with severe other diseases like epilepsy or similar, they need to be diagnosed, and therefore you need EEG. So in other words, it is something which will prevail no matter how the pandemic situation will go on. It's maybe a bit different in research, because in research you are, of course, with the pandemic situation, bound to the typical lab work, and that is done on patients and volunteers, and this is something which at the moment is quite difficult to perform. So For example, in our lab, we had now a lockdown of about 6 months, 7 months or so, where we could not, actually do EEG recordings. Yeah, home office, as you see right now, that is something which we had also. At the moment, it's fine again, but we'll see how it continues. I cross my fingers that it will get all better.
All right, Prof. Okay, due to the time constraint, we will take this one last question from the YouTube chat section. We have from Jennifer Adipal. There you go. Dear Prof, is the electrode commercially available, given this can be request a customized design?
Yeah. The electrodes as such are commercially available, not the current versions, but the previous versions we have had. They are available from a company in Berlin, in Germany. And you can write me an email later on and I can give you the contact details to that company. For research purpose, yes, we also do custom designs. So we do the production of these, we have all the production facilities for these electrodes in-house. So we do the substrates, the coating in our chemical labs. So that is all in-house. So we are able to produce these electrodes in whatever design you wish. That is definitely doable, and for such research questions, yeah, we will simply get in contact and discuss about how we can do what we can do.
All right. All right, I think that should be pretty much for this session. We have come to this end of our Q&A session. Prof. Jens Hauweisen, it is my pleasure to share the virtual stage with you today. Yeah, so it is, hope to see you in someday soon. And with that, ladies and gentlemen, we have come to the end of this session. I would like to invite all participants to join us back at the WebEx platform, as the next parallel session will commence in a short while. So thank you very much to our speaker, Prof. Jens Hauweisen, and all listeners and viewers from our YouTube and also from the Facebook page. See you in the WebEx. See you again in the comments in the parallel session later on. Until then, Assalamualaikum and bye.